It gives me great pleasure to welcome our guest speaker today, Dr. Gavin Jeffries. Gavin is currently the CTO of Fluicell, that you've just heard about, introduced by Mebs. He has a PhD in chemistry from the University of Washington in Seattle, and he was assistant prof at Chalmers University for 4 years and has published over 40 peer-reviewed scientific publications with a total citation count of over 1,000. He's an entrepreneur and founder of 2 biotech and optics companies, inventor of multiple patents and technologies, and he has a strong background in microfluidics, single cell analysis, and optical platform integration. And I'm sure we'll hear more about that today. The title of our presentation today that Gavin's going to be giving you all is Patterning Tissues for Cell Microenvironment Engineering using the Biopixlar Single Cell Resolution Bioprinter. And with that, I'll hand over to yourself, Gavin, and thank you very much for agreeing to come and talk to us today.
So, thank you, Sarah. I'm just making sure I click on the right screen to share. So let's see which screen we're sharing. I am screen sharing. Am I on the correct screen?
Right.
Yep, perfect. There's a little green thing normally around the box, but now I've got a little symbol saying I'm screen sharing. So that's great. Okay, well, thank you very much for this opportunity. I should say good afternoon. We're just 1 hour difference away from you in the UK. We've got a beautiful sunny day here, albeit minus 10 degrees, but it's still beautifully sunny. So we're very happy here to see the sunshine. Thank you very much for this opportunity, Swift Analytical, and also the Tissue and Cell Engineering Society. It's always a pleasure to kind of discuss and communicate about some of the kind of findings and technology to a broader audience. So let me start by kind of having a broader message. I kind of like to set the stage a little bit. As we increase understanding of biology and disease, it kind of becomes a bigger demand or a greater demand for better patient treatments, and this is driving a need to kind of deliver higher patient value solutions, such as those proposed by personalized medicine. And as a lot of us know, an important step within personalized medicine is actually the establishment of human disease models, largely geared towards preclinical drug development. I mean, in essence, what we're looking at is generating targeted treatments with the individual in focus. And this is also connecting. So personalized medicine additionally can bridge the gap towards regenerative and transplantable technologies. So many different technological approaches exist today to try to address these areas. However, bioprinting has emerged as a high potential means of addressing this need for individualized healthcare. So bioprinting technologies have advanced significantly since they started in the early 2000s. These technologies being extrusion-based, droplet-based, light-assisted technologies, for example, all with the goal of capturing the complexity of living tissues, really understanding and be able to replicate, something which not only has the shape of a biological tissue, but also has some of the functions of these tissues. However, this complexity, or reproducing this complexity, especially mimicking the architecture of the cellular microenvironment at the individual cell level, still remains quite a great challenge to this day. Of recent focus, there's been a few recent articles actually describing how to combine these technologies to try to pull on the benefits of each of these different types of bioprinting technologies. However, this requires that these technologies can be freely deployed and freely integrated along other technologies. And that's one of the great challenges. So commonly, these bioprinting approaches focus on generating larger constructs, so to develop tissues. However, to develop a larger construct, you require a support matrix or something to house the cells in. Now, this places the dependency on the matrix, and this matrix can restrict the control over the individual cells that you're actually trying to pattern, and can limit the possibilities for actually modifying the cell's microenvironment after the printing. So once you've got your printed construct, the cells are, let's say, encased in this environment, which is very hard to kind of interact with. But at Fluicell, what we actually do is we invert this idea. So instead of using this matrix, we actually build tissues from the cellular organizational level without using any sort of binding matrix or gel environment. So this focus at the cellular level empowers the investigation of cellular interactions. We can actually see how the cells interact with each other. And this kind of lack of any sort of matrix or any obstacle between the cells leads to rapid tissue development, and also potentially to easier integrating with other bioprinting approaches. The cells are available to be addressed by other technologies. And this also should aid in integration with other common workflows. And what we kind of really focus on is that many focus on the macro, but our niche here at Fluicell is really this micro patterning, so we can build tissues from cellular building blocks. So as was described, my name is Gavin Jeffries, and I'm the CTO here at Fluicell. I'm very pleased to say I've spent almost 20 years now designing approaches to handle and interrogate biology at the individual cell level. So in this presentation, I hope to demonstrate to you how Fluicell employs a technology called hydrodynamically confined flow to build biological tissues from individual cells. I hope to additionally demonstrate to you a breadth of possibilities for patterning tissues and controlling the microenvironment in which these cells are housed. So firstly, I feel I got to explain a little bit on what is hydrodynamic confinement. Well, at its essence, it's controlling the flow of one liquid inside another liquid environment. Now, in practice, what this means is controlling the inflow and outflow at the tip of a very small microfluidic device. And what this does is it generates what we like to call a virtual flow chamber. And this more technically is known as a hydrodynamically confined flow zone. So once you've established this flow zone, it's a very ideal way of delivering solutions to a very small or very localized region of a sample. This is kind of, let's say, this is a little fluidic simulation, I should say, of what it actually looks at. So we have the outflow in the center channel and inflow in the sides. And by maintaining a pressure balance, we can actually establish a steady state flow zone. Maintaining the flow in this manner generates a freestanding liquid probe. And this freestanding liquid probe can then be deployed alongside other technologies. And if we take a look at this under the microscopes, we actually see what really is going on. We can actually see the flow within a very localized region. So on the left here, we'll see actually a microscopic image, and then you should have seen the actual environment at the tip of this device just changing. So this is changing between different colored dye solutions. This is a continuously flowing system, but having this continuous flow allows you to control and manipulate a very small environment, approximately 100 microns is kind of the length of that flow zone. And you can control the content of this virtual flow cell in real time. This is a real-time video that we're seeing here. So as we're focusing at the microscale, we can actually use other technologies which have emerged for controlling things at the microscale. So we can actually embed the power of microfluidics into this device and allow for rapid and very controlled solution exchange. So one recent demonstration of this technology is actually in the localized environment control for neuronal activation of a mosquito stylet. So a focus in particular in this example was how female mosquitoes sense blood, and not only that, but how they differentiate between blood and nectar. So here it's using one of our HCF platforms. This one's called the Biopen. And Vossal Lab at Rockefeller University were able to expose the very tip of this individual stylet to whole human blood. And what this does is it activates the sensory neurons, and they're able to see how these sensory neurons behave to different contents of this whole blood by using different controls and different spiked samples. So if I just play the video here, what we'll see is this hydrodynamically confined zone flows over just the tip of this device. And what we see, or what we saw, is these neurons which go along the sides of this mosquito stylet actually engaging. So I'll just play it again. So what we can see is this zone forms. We see all these red blood cells and other components flowing over the tip, and the neurons along this feeding stylet actually activating in response to those sensory cues. So hopefully, it's very brief, but just to set the stage, I've been able to convey to you some of the kind of ideas and benefits of hydrodynamic flow confinement. at the microscale. So this approach typically is known to enable precision compound delivery. It's really focusing on delivering to just one region and not another one. This also is attached to low volume consumption. We're actually flowing in the nanoliters per second. So the amount of liquid which you're actually flowing is very, very small. And this is done in a contamination-free manner, meaning that once the hydrodynamic flow zone is actually established and maintained, there's no loss to the surrounding environment. So you can actually use this as targeting for an individual part of a larger sample. And all of this gains you local control over a cell or a sample's microenvironment. So the same liquid control principle can be applied to handling cells instead of delivering solutions. So this would enable a means of patterning cells in a completely different style of bioprinting, where we're actually looking at the control and handling of cells at the microscale to build cellular-based biological tissues. But to actually demonstrate this, the easiest way is to actually show you some kind of cool videos and examples of exactly how we do this. So I'll take you through how this approach has been developed, but also how it's been utilized to actually develop these microtissues. So here we have an image of a different HCF device, this time it being one specifically designed for the bioprinter Biopixlar. And if I just start the video, what you'll see is cells actually flowing at the tip of this device. Now they're flowing in this hydrodynamically confined region. As the substrate translates around, nothing is attaching, because what we have here is that the tip is actually being held 250 microns, so a significant distance away from the substrate, to essentially demonstrate that although cells are flowing, nothing is being lost to the environment, nothing is being attached to the surface. Now we're going to take this printhead, we're now going to lower it down to what we would consider the printing region of the sample, so within a few microns. Here it'll be set to approximately 10 microns. And then we'll start this flow again, still confined to the front. But now once we establish the flow, we'll begin to translate the surface. And what you'll see is the surface now captures and holds onto these cells as they exit the device. Now, as the substrate is translated around, this allows the pattern to be formed. So only the kind of cells which are, let's say, exiting the device are attaching to the substrate, but it's only attaching to where the printhead wants them to be. They're not leaving the device area. They're not being spread out to the larger sample area. So it's really confining where these cells are going to be deposited. And all of this can be done in real time. It's got direct microscopy feedback. So you can actually adjust the parameters on the fly to achieve the kind of tissue patterning that you want to get. But if we now apply this printing approach to a little bit more of a recognizable example rather than just a couple of stripes, we can look at a skin histology image. So here, if we take a typical skin histology sample and then we change to an example image where we actually have replicated a very similar structure from individual cells that we can print, and we'll show you exactly how that has been formed. So here you can see the same printing approach where the cells are in this recirculation zone at the tip of the device. The substrate is being moved around. As the substrate is moved around, the cell type is being changed depending on the area and the cell type that is required for that area. And what we end up with is essentially something which very closely resembles the shape and cell kind of distribution that you would see in that histology example. So in this kind of size shape example, what we see is a typical kind of printing approach, or a typical structural approach that we look at a design or take a design of something, the structure of interest, a tumor or a particular cellular arrangement, and then we load the bioprinting device, or namely the printhead, with the cells that are required to build that tissue of interest. We establish our recirculation zone, or hydrodynamically confined flow zone, and then we translate the surface around to replicate that pattern that is desired. What there is, let's say, a base model or a very a starting point which can then be interrogated directly, or it can be cultured or manipulated or have additional things added to this structure. However, what can be done in 2 dimensions can also be transitioned to 3 dimensions. So if we want to build up and establish a multi-layer tissue, this is additionally possible using the same approach. As in 2 dimensions, the flow zone at the tip of the printhead can be translated. But now, as opposed to translating over a surface, now we're translating it over a cell layer. And there's a little kind of cartoon describing how this is done in a step-by-step method, whereby we have a cell layer which is established. We then put down actually a cell attachment agent. This is essentially some components of the ECM to aid in the adhesion between the cell layers. And then we pattern another cell layer on top of this first layer. And effectively, we then alternate between these 2 components, the cell attachment agent and the cell of interest. And by alternating these 2 parameters, what you can do is build up into essentially the Z direction. So establish a 3-dimensional construct. Just a simple example or visual example of what has been done in this was to investigate stem cell cancer cell interactions, where here we actually printed mesenchymal stem cells next to and in contact with neuroblastoma cells, and the very distinctive geometric shape, indicating that we have very high control over where these cells actually end up. So these 2 different cell types were printed on top of a 3rd cell type. So it was actually printed on top of a fibroblast seed layer. This seed layer wasn't labeled, just so you can distinguish between the cell types. But this co-printing kind of setup was established to investigate both the migration and invasion of one cell type into another, and also to investigate the influence of the microenvironment to see if, by tuning the components of what they're actually seated on, or how big these areas are and how that affects these particular parameters. So this cell patterning approach to build up microtissues has been demonstrated for multiple different application fields. We did some multi-cell type tissues, particularly for disease modeling, one being for like a simple skin model using different differentiated states of keratinocytes to essentially establish a very simplistic skin model, but one which we can actually deliver compounds to and actually investigate the interactions or the influence of those particular pharmaceutical compounds. We also have applied it to skin melanoma models to actually build up a melanoma model and actually look at what the size of the cancer area is and how that develops. The single cell types have also been investigated, but with the single cell types, the eye is more towards patterning sensitive cells, towards regenerative medicine applications. So within these areas, we've looked at patterning of stem cells, so mesenchymal stem cells, as well as iPSCs of multiple different origins. We also have looked at patterning very sensitive cells, such as primary extracted neurons, to see how the printing process actually can be utilized in very sensitive and very difficult to handle samples. So I would like to kind of go next and kind of finish on this kind of application train with a specific example. As I kind of elucidated to or touched on at the start, it's understood that we need to have new models or new patient relevant models for various diseases, and particularly for looking at the prediction of drug activity, drug toxicity, but also for the establishment of new therapies, so we can actually have more targeted therapies towards individuals. So to achieve this, we patterned and assembled microtissues using our bioprinting approach, building up the tissues from individual cells. Here in this particular example, we're using 2 cell types. We're using hepatocytes and fibroblasts to actually build up a 3-dimensional structure. So here we used a fibroblast, again, as a base layer, something to have at the bottom of this cellular stack. And then we patterned the HepG2s on top of this base layer. Now, to confine the HepG2s and give them more of a native environment, these were then enrobed with additional fibroblasts to confine them and really help establish a more three-dimensional, or more native microenvironment. But of course, we need to have a comparison against these things. So we essentially established three different models. A very basic model where we looked at patterned hepatocytes, commonly used for a very simple understanding of liver behavior. We then also looked at an intermediate model where we surrounded the fibroblasts or finally the hepatocytes, apologize, with fibroblasts, but still on a standard substrate. And for the more complex model, we actually took this 2D example and actually built that on top of fibroblasts and maintained and grew it in that manner. So once we have these different models established, we obviously wanted to probe the functionality of these printed liver models and liver tissues. And we did this by measuring albumin production, which is a common specific marker for hepatic cell metabolic activity. But within this study, what we did is we took each of the printed tissues, where each one was actually replicated 4 times, so that we have enough signal to actually have the further processing. So we had these substrates with 4 replicas of each model on, and these were grown for 7 days. So we cultured them for 7 days, and then after 7 days, we sampled the cell-free culture supernatant, and we took this supernatant and actually performed ELISA assays on it to quantify essentially the amount of albumin which has been secreted by these hepatocytes during this kind of time process. And what we actually saw is highlighted in the graph in the middle, in that we actually saw an increased albumin secretion in the more three-dimensional models. Now, the kind of most apparent observation is that at least these cells are producing albumin much faster and probably with superior behavior when they're compared to just the standard monoculture or a simplistic 2D model. So in conclusion with this, what I hope I've been able to show you today is just a couple of the capabilities of actually building tissues using cells as the building blocks. So this can be done in both 2D and 3D, depending on its need. And also, due to the kind of freestanding nature of the technology, this can be done on multiple different types of substrates. The cells could be housed and maintained in growth media. We don't need to use any sort of gel matrix. We're actually positioning the cells onto a surface. And this, at least we believe, plays a role in the cell viability during all of our viability assays, which we have performed. And we see very similar cell viabilities to that which would be seen in cell culture. As there is nothing really stopping the cells from reaching out and interacting with each other in these printed constructs, the cells actually establish connections very fast. And more often than not, we actually start to establish confluent tissues in as little as 24 hours. So the cell-to-cell communication is actually very rapid. And I just want to wrap up with a few of the messages that I kind of hope that you can take away with you today by bioprinting with the cells at the focus. So microscale control allows for patterning tissues at the level of individual cells. You can actually do this by looking at the cells themselves. And these cells can be maintained in growth media throughout the process. Now, this has the benefit that it is then compatible with most conventional cell handling and culturing methods. There's no real need to change workflow. This can fit into most people's current workflow schemes. But this also aids in the next point, which is that nothing is really shielding the cells. These tissues are interfaceable directly. So you can actually interact with the cells, or you can change their microenvironment, as we were showing with early on with the HCF demonstration. You can deliver compounds directly to parts of the tissues that you're most interested in. But because this is at its core a microfluidic technology, it has a huge advantage in that it's perfect for handling scarce and valuable cell sources, as the volumes that are consumed are typically in the few microliters. So thank you for listening to me today. And I hope, even though we had some technical difficulties, I was able to convey to you kind of some food for thought and a different way of actually establishing microtissues by building using cells as the building blocks. And I welcome any questions that you may have.
Thank you so much, Gavin. It was a really presentation on a really interesting approach in how to construct these hierarchical tissues. I think it'd be of great value to all of our delegates here today. As I mentioned earlier, we're going to be using the Q&A section, please, for posting your chats. We have a few questions that have already come in, so I'm going to start going through those if that's okay, Gavin.
Yeah.
So Michael's asking, can you please comment on the requirement of individual cell level microenvironment, oxygen, pH, and glucose sensing for 3D tissue engineering applications?
So I'm trying to see where that one is in the list. Sorry. Okay, the very top one. Can please comment on the requirement of individual cell level oxygen, pH, and glucose sensing for 3D? I think, well, yeah, I don't know if I've clicked the right button there, Jonas, but hopefully you can handle the administration on that one. When we're looking at the very small areas, obviously you do want to control the environment as closely as possible. When we're building, at the cellular level, because we don't use any matrix or anything, we can directly deliver whatever kind of compounds we want just in the media. We don't have to deal with diffusion that you would normally have to deal with if you bioprinted in a gel construct, for example, where you would have to really pay attention to if there is a buildup of weight products or change of pH or things like that. We could, if we wanted to place sensors, in the cells, we could use, direct sensors inside. But for the most part, our microtissues actually will resemble very closely how it would be in culture, it being just a few in 3 dimensions, it would be just a few cell layers thick. So as long as it's not very thick, I think it would be handled exactly the same way that you would handle it in a cell culture.
Thank you, Gavin. The next question is from you. You asked, what is the time frame for the liquid exchange of the biopen, and what is the surface area covered by the liquid at the tip?
Okay, so the time frame for essentially the switching time, it can be very short. So it really depends on the pressures that your sample is willing to take. More often than not, we're in the I would say for standard biological experiments in the sub 200 millisecond solution exchange time. So it's significantly sub-second. It's possible to go down closer to 100 millisecond range, but more often than not, because of certain flow constraints that you don't want to flow that fast, close to your cells of interest, you'll typically be doing it in a couple of 100 milliseconds. With regards to the kind of exposed area of the sample, under standard operation, most HCF style devices that we make are exposing approximately 100 microns wide, as in and about 80 microns kind of extended into the sample. So it's actually quite a small region, normally enough to cover a few standard size cells.
Thank you. So Evo has a cell biology and is asking and says that although the concept sounds great, has a question about layering each cell with a skin model. For example, will it be great to make an immature model of undifferentiated cells and let it mature?
Yeah.
Printing the external environment to enhance self-assembly of the cell would be best. Would this be possible with your system, or is it only for printing the cells?
So you absolutely can change the microenvironment. It really comes down to what the response time is. So typical exposures as typical environmental changes that we do are for triggering things such as ion channel experiments or for delivering drug compounds to cells of interest. This is done for seconds to minutes at most. Theoretically, this could be maintained for an hour or two without really any problems. But I think from my understanding of when we work with the keratinocytes, you would need to be modifying it for quite a while to actually do that. So when we built our kind of skin models, we actually took the different cell types, exposed them to the different compounds to change how they were growing, and then once we're sufficiently happy they were now the cell type that we wanted, we then lifted the cells, loaded them into the print head, and actually printed them in their already established state.
Thank you. So quite a quick question from Edward, who asked about whether or not you can adjust the nozzle size to print spheroids rather than just...
It's a very good question. It's something which has been approached by people. I think the easiest thing to state currently is that we do not have anything which operates on that size scale. Although spheroid work is very interesting, I'm not entirely sure of basically what would be possible in the future with regards to that. It has been raised, and it is a very interesting idea to use spheroids as the building blocks instead of individual cells.
Thank you. So Journey has a question which is more about, I think, the business model. So I don't know if it's Gavin or Mebs that wants to answer this one. And asks, what made such a huge player like Hoffmann La Roche cooperate with such a smaller player like Fluicell?
I think what is I'd say unique with Fluicell is that we have the company is built around this kind of academic idea of empowering life science research, and it's really got this kind of empowering researchers to achieve their goals. I mean, that's really what we want to do. We want to drive, the kind of the science and as well as the technology forward. And along that journey, we do have, let's say, a quite nice, reputation of we do understand how to handle and work on this type of size scale, and how to work with flows as well as biological cells in this area. So with that kind of in mind, the size of the company outweighs the kind of uniqueness that we bring to the table, in that we are an empowering kind of technology company, and we do have kind of a nice support team of people which not only have a background in science, but also in technology development.
Yeah, that makes sense. Thank you. It's very important in the innovation pipeline, isn't it, really, to have all the different sizes of companies working together and different expertise.
Yeah.
We've asked another question. If you have an induced pluripotent stem cell reporter cell with specific cell markers, for example, P63, the basal keratinocytes, coupled with a fluorescent tag, maybe you can differentiate the iPSCs into a mixed population of skin cells, from which you can use the fluorescent reporters and your own method to lay the cells in a perfect way to obtain an actual skin tissue from stem cells. Do you think that this approach will be achievable, or is it something that's too complicated to investigate right now?
So I'm trying to see exactly. So I'm trying to reword it to make sure I understand exactly what the question is. So if you have a specific marker, so we have a protein marker, you can differentiate the iPSC into a mixed population. Yeah, that's possible. Your method to layer the cells in a perfect way to obtain actual skin cell. Do you think it's appropriately achievable? I'm not entirely sure of the question. I would say that I don't want to say anything wrong. So what I would do is if at the end you see, you'll see both our email and Swift Analytical's email, maybe you can elaborate a little bit, Ivo, on the question, so I can answer it properly, because I'm not quite gaining exactly what you want to do, and I don't want to give you an answer which is incorrect. So maybe we can take that one after.
We'll try and do that. I'm just conscious of time. We do have another few minutes. We've got quite a few questions to come through. There's an opportunity to take some of these questions offline as well. So either if this is something you're really excited about and has potential for you, we'll make sure we get back to you, okay? Donovan says, does the print head have multiple extrusion channels? For example, in the model you showed, can the 3 different cell types plus the adhesion layers be printed in a single print?
Yes. So they're typically the 3 channels that you see, one is outflow, 2 are inflow, but we load 4 different samples into the print heads. Generally, what we would do is we would save one as a washing solution just in case, but yeah, we absolutely can do 3 different cell types and a printing agent at the same time.
Thank you. We have a couple of more questions from Yuha, who asked, the first one is, can you give an indication of how many cells are required for printing? Is it millions of cells, squared or something similar? Or?
So in general, so we've had very successful printing runs on very few numbers of cells. So if you look at our most basic model, which it's like a single square of hepatocytes, for example. So it's like a 500 micron square. There are a few hundred cells, so between 600 cells and 1,000 cells, as long as they're close to each other in a culture plate and it's handled correctly, they will grow perfectly fine. You don't need to have that many cells actually attached to the substrate.
You have also asked, what are the CAAs and can they influence the cell?
So essentially the CAAs, it's a cell attachment agent. So because to actually physically stick one cell on top of another, you need some sort of encouragement to do that. So depending on the cell type, it changes, but essentially they are components of like either ECM or media. So like attachment proteins such as things that you would use to attach to a surface like laminin or polyorthenone or polylysine and other kinds of adhesive, either amino acids or proteins. But we vary it depending on the cell type, because each different cell type kind of prefers a different adhesive agent.
Thank you. Our next question is from one of our delegates who asks, I just lost that question. Sorry. Sorry, I was in the middle of it, but it's just disappeared. The next question I have here is Monoha, who asks if in the 3D printed tissues, if the 3D printed tissues need to culture for a certain amount of days to develop their own innate extracellular matrix in consideration to
drug screening. Yeah. So when we're building it in 3 dimensions, generally what we do is we like to give at least overnight, if not 24 hours between each layer, so that they can really, establish kind of a level of communication and confluency as we're kind of building up. So with that regards, I would say, yes, we would probably give it 24 hours afterwards if we wanted to make sure that it's, a fully interacted kind of tissue structure.
Shahid says, thank you for the amazing presentation. Shahid's question is, can this approach be applied into the recellularization of decellularized scaffolds, specifically kidney scaffolds?
So I think what it would largely come down to is what the scaffold looked like. I mean, one of the things that is very good with this type of technology is that because everything happens at the tip of a device, and this tip of the device can be moved in 3 dimensions, arguably then, as long as the cells will kind of stick or be captured by the surface, then you could overprint onto anything as long as you can translate around the objects quite freely. The other kind of caveat would be that it is hydrodynamic confined, so we need to have it submersed in a medium. We can't do this in air. This is in the same way that we print typically in culture media. Culture media is what's on the substrate. It's also what the cells are housed in. So we need to have this kind of all liquid environment for actually doing the printing.
So we have a question on what are the limitations using hydrodynamic flow confinement?
I think the biggest one is probably size. I mean, this type of technology can be multiplexed, so you can have many of the same thing. But this is based on like laminar flow technology. So laminar flow needs to be maintained. So I mean, you can't go like many millimeters, for example. I mean, at some point, the kind of fluid physics breaks down. We really focus on seeing and patterning the individual cells, and if we kind of keep that having the cells as the focus, then the limitation, as we would see it, would be that we need to have the cells attaching to the surface in these HCFs, essentially a limitation.
Thank you. Deepak asks, have you profiled the cell adhesions, such as integrins, which can play a pivotal role in biodirectional signaling? What influence would this have on transcriptional pathways and in turn protein secretion?
Yeah, it's a very good question. We haven't done those profiles yet. It is looking at the different roles and actually looking to see if any of these components are actually, integrated or absorbed, or if they're, retained in the final structure is something that we plan on looking at in the future, but we haven't profiled it yet.
You have asked again, are there plans to miniaturize the BioPen to multi-well applications like MEA platform?
I'm not sure what an MEA platform is, but typically the kind of HCF tech that we have. So we have 3 different platforms that we currently have. So the BioPen, the BioZone, which is going to be released soon, and the Biopixlar. They can go into multi-well platforms. Currently, we don't normally go above 12, like a 12-well plate. Going significantly beyond that, I think is outside of the current, not the technology's limitations, but the fabrication limitations to make, tips that sharp and that kind of flexible. But we do have the ability to go to, I think, 12-well plates currently.
Thank you. Reem says, in your system, are you able to control cell differentiation, dedifferentiation, and transdifferentiation, or do you depend on printing the cell types that you require in your layers?
Yeah, we generally depend on, so if we want different differentiation states, then we'll generally pre-differentiate the different states and then we'll load them. However, anything which can be triggered or done, let's say quickly, as in within, if it's a stimulation which only is required for a few minutes or an hour or two, then that could be done in place afterwards. So you could print the construct and then expose part or a large section of the printed tissue to a particular trigger or a particular differentiation trigger compound to actually drive differentiation of one part or all of it. But it would require that if we're doing it after the fact, that the exposure time could be within minutes to an hour or two. Longer than a couple of hours is quite challenging to maintain.
Davide asked, do you think this technology could be used to replace animal models by producing small biological viable tissue models, for example, with skin to study wound healing?
Yes, I mean, it's definitely one of the areas that we have in focus. The big advantage being that you can use, let's say, human stem cells or differentiated stem cells to actually be, used or design human tissue instead of animal tissue. The kind of, let's say, big advantage of doing it in that way is that you can generate fairly reproducible samples. So you can have many duplicates using exactly the same cell origin. So you don't have to, have as many kind of controls to state, okay, this can be part of this population, because you can actually say what the genetic origin of all of these tissues were and exactly how they were built and what all the shapes are. So I think that in the future, that kind of direction of minimizing animal usage, I mean, it's always going to be there, but this type of bioprinting, I think bioprinting in general is really has in focus of minimizing the use of animals and actually using more human specific kind of tissue models.
Thank you. Gavin, your talk has been very, very popular. You've got lots more questions in this. I'm afraid we're running out of time. So what we'll do is we'll stop answering the questions live, but I know that we're taking a note of all the Q&A questions that have come in, and we'll endeavour to get back to the delegates who've asked those. We will make the recording available to everyone and everybody else who hasn't been able to make it today that we're interested in watching it. I'd like to thank you again, Gavin. Really exciting talk.
My pleasure.
I think that some of the examples that you've shown, particularly in the bioengineering on the liver model, leads me quite nicely to inform you if you could possibly, well, we've got the contact information there and the Swift Analytical, those can make a note of. But if you possibly stop sharing screen, because I just want to show one.
Yep, absolutely. No, not that one. Sorry, I need to stop the share. That one. Yes.
So it leads me quite nicely to introduce the next virtual seminar, part of the seminar series, which will be next month. So that's taking part on Thursday, the 10th of March at 10 o'clock in the UK. And this is going to be Dr. Luca Urbani and Lola Ajayi from the Institute of Hepatology Foundation for Liver Research, who will be telling us about the excellular matrix liver disease, liver cancer, and bioengineering for disease modelling. So those of you that can make it, please do come along. Find out you've got the contact information for Swift Analytical. I'm sure you want to go there and find out a lot more information from them, but also Tissue and Cell Engineering Society. For those of you that are not familiar with the society, we do an awful lot in this area. It'd be fantastic to have you involved if you're not already.